All right, welcome to our demonstration video and assistance video for your Meiosis Models Lab activity. So, in class, we finished this pre-lab together. If you didn't get this part, it is posted in the assignment in Canvas. So, go get this caught up so you're ready for the actual lab activity portion. For the lab activity portion, we need the back of this page. So, our materials list. You guys need 2 long pipe cleaners of color 1, 2 long pipe cleaners color 2. It does not matter what colors you pick of the ones available. It matters that you pick 2 different colors and you have 2 pipe cleaners. So, for example, I might pick color orange, 2 of them, as my first color, and maybe I pick green as my second color. There's 2 for color 1 long, 2 for color 2 long. I need 2 short pipe cleaners color 1, 2 short pipe cleaners color 2. These are my long pipe cleaners. I need 2 the same colors in the short versions of them. So here is 2 orange, and here is 2 green, all short. Then we need 2 colored pencils to match the pipe cleaners. So if this was what I was using, I would need an orange and a green colored pencil. We need scissors. We are going to have to do a tiny bit of cutting. And then 2 circle papers. For you guys, today in this demo video, I'm going to be using my circle papers. Your guys' papers will be out and attached to the lab station tops for you guys to already be out and use, so you don't have to worry about that part of your materials. For today's video, because I want to know that I used different colors than you guys are going to be using in class, I'm going to use some different pipe cleaners. My color 1 is going to be silver, and gold will be my color 2. I have them in long and short versions. And instead of colored pencils, because I don't actually have gold or silver, I'm going to use Sharpies for my drawing. Okay? Everything else is going to be the same. I'm not going to go all the way through the entire activity, but I'm going to get you started and do a couple of the phases. So, in honesty, it's pretty simple. Meiosis 1. Follow the directions to model and draw each phase. Even if I forgot to tell you to draw what you modeled, draw what you modeled. So, interphase. The cell is growing and the DNA is replicating. The DNA looks like a ball of string and has not yet formed chromosomes. So, it's kind of just a big mess. During interphase, replicate your DNA by adding an identical pipe cleaner to each. Twist them together to make an X. Draw what chromosomes in interphase look like in the box to the right. So, I have my 2 small silvers. I'm going to add these together, twist several times so they stay, and make an X. I'm going to do the same for my long silver. Twist so they stay, make an X. My short gold, twist, make an X. And this is interphase, so long gold, twist, make an X. Nothing is organized, really just a jumble. So when I draw this, I will have a jumble of long and maybe my short, and my long and short. Model, drawing. Then, prophase 1. During this phase, the chromosomes form and chromosomes pair up. Crossing over occurs. The spindle fibers attach to the centromeres. Place your homologous chromosomes together on the cell. So, that's this thing. We're going to use one circle. Remember, homologous means same, size or relative shape. So my big X's go together, my little X's go together. Make your chromosomes cross over by cutting and trading pieces. Draw your finished model after crossing over by placing your paired homologous chromosomes on the cell. So here's where my scissors come in. My large chromosomes are going to trade bits of genes. So I'm going to take some sections with them together and cut them. This silver section gets to trade and twist on nice and tight so we don't lose it during our model to the gold. And the gold goes to the silver. And these two have crossed over. They're kind of just chilling wherever they want in the cell. Same thing could happen with my small chromosomes. If you are very confident in your cutting and twisting abilities for pipe cleaners, you can cut a leg and go. You don't have to if you can't actually get things to stay. So this is prophase. Things are still jumbled, crossing over has happened. So now we get to draw this in our box. So I'm going to start with silver, and I have a small square and I have a big, that's an X, not a square, big X here that has one gold leg. Connecting with it, I have another X that's gold with a silver leg. And my little one has a gold X overlapping. So my picture I drew looks like my model. Then I just keep going. I read metaphase and say, learn what happens here. I place my homologous chromosomes together in a line on the metaphase plate. So metaphase, they line up. They're still in their pairs. In the middle, I make my model. I draw it. I read the next piece. Go to the next side and we just keep going. Telophase 1. 2 new cells begin to form by pinching apart. The spindle fibers are pulling the cell apart and 2 new nuclei are forming. So this box becomes essentially 2 new cells. Model your 2 cells beginning to pinch apart. The homologous chromosomes are now moving to separate cells. So now they're starting to move and I'm getting into the other side. And then I draw what I modeled. So I can model and draw what I've got. This and so I modeled it, I drew it. Cytokinesis, the cells completely divide. Notice that the 2 new cells have gone through a reduction division. So they reduced and divided how many chromosomes there were. There is now only 1 of each homologous chromosome in each cell. Model the 2 cells completely There is no organization. It is kind of just full disarray. They're floating all over the place. So I now have 2 new cells. I modeled it, I drew it. Then just keep going. Prophase, well, I know I have 2 new cells. So I'm actually going to divide each of these boxes because I'm never going to go back to 1 cell. I'll always have at least 2. Anaphase, the sister chromatids separated and pulled apart to opposite ends of the cells. So now we get to untwist the X. Don't break off the leg, but we untwist. I now have 2 sister chromatids that are going to start getting pulled opposite directions. Things start getting pulled away. This, anaphase. So I'm going to draw it here on anaphase. Then telophase 2 and cytokinesis. The sister chromatids separated and pulled apart to opposite ends of the cell. Model the 2 sets of cells begin to pinch apart with 4 nuclei beginning to form. Now is when you need your second set of circles. So this, 2 of these sister chromatids are going to get pulled into this cell, and 2 of these sister chromatid are going to get pulled into this cell. Now I have a small chromosome and a large chromosome, a small chromosome and a large chromosome, and the same in these 2. So I would draw what's inside these 4 in my telophase 2. So I actually need 4 boxes. And then let's draw this. So that's these 2. Then in my other 2, that is the end of my models. Remember, this is a video, so if it didn't make sense, play, pause, repeat, rewind as many times as you need to help you do your own model.